Thanks very much, Karen. It's a real pleasure to be here today. So thanks so much for the invitation. So my topic is how clinical trials are designed and why there are more clinical trials in IgAN now. So first of all, I thought I'd start off what's the goal or the purpose of a clinical drug development program, and it's really to generate compelling evidence that a drug is effective at treating a disease, and we call this efficacy. Determine what the side effects are, and we describe that as safety and tolerability. And then importantly, to demonstrate that the benefits of the drug outweigh the risks. And so we'll have to form a benefit risk assessment. And we hope that if we achieve the above, that we can get approval of the drug for doctors to prescribe to patients. And as John mentioned, in the UK, it's the MHRA, or the Medicines and Healthcare Products Regulatory Agency, which are going to determine whether a drug is approved or not. If we think about a clinical drug development pathway, we've got three main phases. We've got phase one. These are generally performed on healthy volunteers, and the purpose of the study is to understand safety and tolerability. And these won't be large studies, but there will be a pathway of understanding safety and tolerability to enable us to go to the next stage, which will be phase two, where we'll start testing drugs on patients with the knowledge of that safety data from the phase one. The purpose of these will be safety, and also to understand whether the drug has some benefit in terms of efficacy, but what doses are required. And so this is really a dose finding time for phase of development. Then once we've found the dose and the duration of treatment, then we move into phase three, where there'll be larger studies, and we're really confirming the benefit which we've from in terms of safety and efficacy in a large population on the dosing regimen selected from phase two. So looking at a little bit more detail, we come to phase one. So these are typically described as first in human studies, and they'll take place in a dedicated facility called a phase one unit, and studies on healthy volunteers, and they'll be monitored 24/7 while they receive treatment. And most studies are divided up into two parts, and the first part, called a single ascending dose, or a SAD study in clinical development, we really do like our acronyms. So this is called a SAD study. Doesn't sound very good, but that's and we start with cohorts of patients, and we start with a very low dose. Again, I mentioned these are healthy volunteers, so they'll just receive a cohort of healthy volunteers will receive a single dose, and we'll measure to see whether there are any side effects. And we call these side effects adverse events, or AEs. And if there are no AEs in the cohort one, we'll feel safe to then go on to cohort two, where we can increase the amount of dose which the subject receives. If that looks good, we'll go on to a higher dose again, and we'll carry on until we start to see side effects or adverse events occurring. Once we've conducted and we've found a sort of where the adverse events start occurring, we can start thinking then about the second part of the study, and this is the multiple ascending dose, so the MAD part of the study. So we'll choose a dose from part A, and the purpose here is to give multiple doses. So a patient, for example, or a healthy volunteer, will receive one dose every day for 7 days. And again, we'll look to see side effects, and if none occur, then we can actually try 2 doses every day. And we'll carry on this. And this says to understand where really does it start getting side effects occurring, which may limit how high we can dose. And the purpose of this is really to inform on the dose starting for our phase two. But we'll also try to understand how the drug behaves in the body as well, and how quickly it gets broken down. So this is often called pharmacokinetics, or PK as we call it. This is an important aspect of phase one studies. So then we come to phase two. So we've learned about safety and tolerability from our phase one, and the purpose of phase two is really to find a dose regimen that's effective and safe in patients. Often studies will range from somewhere between 50 to 200 patients. And one of the questions asked is, how does it compare testing a low dose or a medium dose or a high dose versus a placebo? But importantly as well is how long do we need to treat to get an effect of the drug in patients. So we might have a short duration, a medium or long duration. So these important parameters, which in this sort of phase 2 phase, which will enable us to choose the most effective and safe dose to go into phase 3. And these are to confirm those findings in that dosing regimen and generate sufficient data to support approval of the drug. Now, regulatory agencies may require 2 or 3 of these studies, and in large indications, large diseases, might require these studies maybe 1,000 patients or 5,000 or even 10,000 patients. But in rarer diseases, sometimes only 1 study is required, and there's some leeway in terms of the number of patients which need to be treated. So these are the 3 phases which we have in the clinical development. And we hope that if we show good benefit risk profile, the drug has the potential to be approved. So when we're considering a phase 3 trial, there are different groups which we have to take into account were important in considering the trial. So first of all, obviously we have the patients. When we design a phase 3 trial, very much have to have you in mind. We have to make a study which is attractive and interesting to you, so that you're willing to participate. Without you, there is no drug development. There are no trials. So it has to be compelling to you to participate. And equally, the study has to be compelling to doctors. There has to be a prospect of providing valuable data in the treatment of disease. Then we have regulatory agencies, and we have to ensure that the measurements that we're making and the results which we're going to generate are in line with what they need to see to be able to approve a product. We have research ethics committees. Now, these will the purpose of these is to safeguard the rights and the safety and the well-being of patients in the study. And ultimately as well, for phase 3 studies, we have market access organisations, such as in the UK, it's NICE, the National Institute of Health and Care Excellence. And we need to show that a drug, a new medicine, is cost effective and to be made available, a decision for them to be able to make available for that drug to be allowed to be prescribed under the NHS. And then we also have a pharma company, and they're overall responsible for the trial, and of course they will be financing it. So these are really the main, not all of the stakeholders involved, but many of them. So if we think now, what are the key elements to design a phase 3 trial? When we think about, so, well, first of all, the patient population. And we devise in our study protocol inclusion-exclusion criteria. And the purpose of this is to ensure that the patients we enroll, they have the disease which we intend to treat. They may benefit from the drug, not put at undue risk. And they're also, the patients are likely to complete the study as intended. And we assess these eligibility criteria, the inclusion-exclusion criteria, what we call a screening process. And once we've screened patients, then the next decision is really to understand what treatment regimen is are going to be received. So in other words, we've got our investigational drug. We've understood what dose and what from the phase 2, so we get information from that, and also the duration of treatment. But we also need to understand what comparator what are we going to compare to? So it could be a placebo, or it could be the current best treatment for the disease. I mean, we would wish to show that our investigational drug is better than the best treatment currently available. Then another aspect of a trial is randomization. So in other words, we take our screened patients and then we randomize them to either receive an investigational drug or a comparator drug. So why do we randomize? Well, the purpose of this is to ensure that we don't bias the studies. There's no sort of favoritism over which treatment a patient is going to receive. And this is important, a really important aspect of a trial to ensure that we don't bias it and we can make a fair assessment. And another aspect which we bring in is called double blinding. And a double blinding is ensuring that neither the doctor nor the patient, and indeed nor the pharma company, know who's on which treatment. So this ensures that everyone's going to get treated equally. And again, there's no biases introduced. If we knew that a patient was receiving one treatment versus another, they could end up being treated slightly differently or behave slightly differently. And we want to try to minimize that and ensure that no bias is introduced into the study, and we can really make a fair assessment of whether the investigational drug is better than the comparator drug. Then we come to endpoints, and endpoints are really critical. This is what parameters are we using to measure safety and tolerability, and also, as important, to show that the drug is effective. So what are we going to measure in particular? And we call effectiveness efficaciousness of drugs. And so these endpoints, they're typically measured at the end of the study once all patients have completed the treatment phase of the study. And then we've also got the number of patients. So once we've chosen our endpoints, what we're going to measure, then we can decide and use statistical methods to estimate how many patients we need to show a benefit for the investigational drug versus the comparator drug. Endpoints, assessments, and visit schedules, for all of these, we will consult patients and doctors to make sure that the study collects the right data to evaluate safety and tolerability. The measures of efficacy, the endpoints are meaningful, they mean something to patients and doctors. And we have to make sure that the study is practical and not overly burdensome to you. In the past, I have to say, sponsors or drug companies were very, very poor at consulting patients, what patients need. But this has changed drastically over the last 10 years. And now there's far more emphasis in bringing both patients and doctors together to understand their needs for the trial. And we typically have advisory boards where we bring groups of people together to discuss the protocol and make decisions on what it's going to look like. But equally, endpoints, we have to make sure the endpoints meet the needs of regulatory agencies. They're the ones who are going to be decided on drug approval. There's no point having endpoints in a phase 3 study designed to get approval if we don't address what their requirements are. So in the UK, that's going to be the MHRA. And then also we need to consult the market access organizations, so NICE, to ensure that the endpoints are meaningful from their perspective, and that they will be able to make their assessments to ensure that we have adequate data for them to put it on the formulary for the NHS. So when I started in drug development in IgA nephropathy, this goes back all the way to 2010, and I was responsible for the clinical development program for Nefecon, which has now recently got approved. And we were the only company developing a drug in IgA nephropathy. 2017, around about 10 companies. Now, as you saw from John, there's over 20 drugs in development. It's a massive change. It's unbelievable. And that picture which John shows of the desert-like picture to where we are now, and it's just astonishing. So why? How's this come to be? Well, it's all about endpoints. Everything's to do with endpoints here. So back sort of 15 years ago or so, now for a drug to be approved, we really need to be looking at endpoints of kidney failure. And that's what the regulatory agencies, they required. So in other words, what do we mean here? It means that if we conduct a trial, we have to show that less patients progress to kidney failure than in those treated with a drug versus a comparator. IgA nephropathy, it's a disease which generally patients progress over many years. It's not something where a patient will get diagnosed and within 1 year they're going to reach kidney failure. So this means that studies take a long time, and actually we require a lot of patients. And this put drug companies off actually developing. We have to spend a long time conducting the trials, high risk, high cost, and it was just an unattractive proposition. things started changing at about 2012 to 2016, and suddenly regulatory agencies start to allow change in GFR as an approvable endpoint. In other words, the rate of loss of kidney function. So if we could slow the loss of kidney function in those treated with drug versus comparator, that could be approvable endpoint for a drug. And smaller studies required more 300 to 500 patients here, 3 to 4 years, a trial B, probably more than 4 years. But this was becoming acceptable now, or at least under consideration. But the big change really was regulatory agencies allowing a reduction in proteinuria as an endpoint for conditional approval. And I'll come back to what conditional approval means. But here what we're trying to do is show that there's going to be a greater reduction in proteinuria in those treated with a drug versus a comparator. And proteinuria changes when we treat patients with new drugs, they can occur quite rapidly. So we don't need the trials to be quite as long, and we can in fewer numbers of patients. And suddenly the opening up of the change in EGFR, and in particular the proteinuria, caused the sea change, which we see now in the number of drugs which are being developed in IgA. So unbelievably exciting times. So actually, what happened to cause regulatory agencies to change their mind about endpoints here? So here's a little schematic. So the first sort of diet or figure in the middle, that's representing kidney function. So when kidney function goes down, we know that there's a risk of patients progressing to kidney failure. And indeed, if you have proteinuria, high levels of proteinuria, and the greater the amount of proteinuria, the higher risk you are of progressing of having rapid decline in kidney function. And indeed, as well, there's the same association or correlation between high levels of proteinuria and the risk of renal failure. So, however, some statistical meta-analyses were conducted on data from historical IgA nephropathy studies. So in other words, lots of randomized controlled trials have been conducted in the past, and there was a gathering of all of this data together to analyze it in this so-called meta-analysis. What this showed, meta-analysis, was really important findings. So first of all, here we have a patient or a population who are progressing, have rapidly progressing IgA nephropathy, so there's a decline in kidney function. Now, if we slow that decline of kidney function, then it predicts that there's going to be a reduced risk of kidney failure. You're less likely to end up with kidney failure. The next point was, what happens if we reduce proteinuria? So we lower proteinuria. That was shown then, lowering proteinuria predicts the slowing down of loss of kidney function. And indeed, the final arm was reducing proteinuria predicts the reduced risk of kidney failure. So showing definitively and understanding the mathematical relationship between these variables gave regulatory agents the confidence to say that we could use proteinuria as a marker, or reduction in protein as a marker for conditional approval, and also rate of loss of EGFR as an endpoint for full approval. So what happens now in a typical phase 3 study? So how does it look? So we'll be screening patients, and we have our investigational drug and our comparator drug. We'll need to have these. So we'll be looking at a study which are around about 4 years. And a typical study will involve, phase 3 study will involve 300 to 400 patients, and we'll probably need to recruit them, patients at around about 150 hospitals, around that number. They'll often be conducted all around the globe. And so we'll allow for 2 years for patient recruitment, and then 2 years typically for patient participation. And we'll look to evaluate proteinuria at 6 to 9 months after treatment. And we'll measure rate of loss of EGFR at 24 months. So we'll look at see how the slope of EGFR over time. And the great thing about having this early readout is that it can provide a go, no go decision point. So it means that we won't, if there's no effect of the drug there, we can stop the trial early, and that which will mean ensuring that we don't expose patients unnecessarily to an investigational drug. However, if we do show benefit in proteinuria, there's an opportunity to apply for conditional approval. And this is exactly what has happened to Nefecon or Tarpeyo, which is now has a conditional approval in both the US and in Europe. But there comes a commitment to conditional approval. It's only conditional. It's on the condition that we continue the trial and will show long-term efficacy and safety. And that's where we will have to show the benefit on eGFR at 24 months to remain the drug to remain approved. So that's to provide, that data will provide full approval of the drug. And there are about at least 5 trials ongoing which have this basic core design to them. And we have, as John mentioned, we have Nefecon or Tarpeyo, which has now generated that 6 to 9 month data on proteinuria. We still wait for the eGFR data, but it's got conditional approval. We have Sparsentan, which is the stage now of applying for conditional approval in the US, and that will also come in later this year in Europe. And then we have other drugs which are close to readouts for their proteinuria readout. So it's unbelievably exciting times, and it's a, it's a sea change. We hope that these drugs, many get approved, and it'll be a sea change to the lives of people with IgA nephropathy. So that was my last slide, and I was wondering if people have any questions.